what we are able to do is go into these samples and interrogate them with modern technologies, like proteomics. You can look at 7,000 proteins now in a human blood sample, thousands of metabolites, right? And just ask this big sort of data first question: what's changing with aging, of course, but also what predicts the future?
Aging. Like gravity, it pulls on each of us. Why do some of us age without illness? How do our bodies and minds experience aging at the cellular and molecular level? What's the future of aging in our society? And maybe, most importantly, what can we do about it today? My name is Gordon Lithgow, and here at the Buck Institute in California, my colleagues and I are searching for and actually finding answers to these questions and many more. On this podcast, we discuss and discover the future of aging with some of the brightest scientific stars on the planet. We're not getting any younger, yet. Hi everyone, welcome to the show. Today I'm delighted to have Kristen Fortney on, and I've known Kristen for quite a few years now, all the way back to her days at Stanford when she was a postdoc. Kristen was really quite unique in coming out of that postdoc in the sense that she immediately realized the potential for the aging research space and was also very entrepreneurial and wanted to go ahead and get in there and form a company. Kristen is the CEO and co-founder of BioAge. Kristen's company uses human samples and health records to develop targets for interventions. Kristen has been very much in the eye of the scientific community as a shining example of how we can move from the basic science into a successful startup company and beyond. Welcome, Kristen.
Thanks, Gordon, for having me. It's always great to chat with you.
You know, we know you as one of the superstars of the aging community in biotech. I mean, in all the scope of biology, botany and marine science and everything else, how did aging come to be the thing that fascinated you?
So personally, I've never been, I guess, a science for science's sake person. It's more sort of like, what can we do with these tools? And so biology and its impact, and medicines, right? And the impact on healthcare were always very exciting to me. And aging, like, as you know, right? Like, it underlies so many different diseases. It's like a master key in some ways that can unlock a lot of different benefits in human populations. And it's also very understudied, right? Like, there's a lot of really exciting new work to be done there.
So BioAge Lab is really based on your computational biology experience. And at the outset, I guess you had some strong ideas about how to find targets for interventions in aging. So maybe you can talk about that.
Yeah, so zooming out a little bit, if you look at like the kind of data sets that have been really transformative for biotech in general in the last 20 years, and it's the right human data sets, right? So the large biobanks like UK Biobank, like Decode in Iceland, which Amgen bought a few years ago, and mapping out the different molecular pathways in large human cohorts, the different genetics in large human cohorts. I think, people have shown that if a target has what they call genetic evidence in a human population, it's much more likely to be successful in the clinic. And this saves so much time and so much money. And part of the rationale and the impetus for BioAge is like, what's the version of that human data for aging? and if anything, I feel like it's even more sorely needed in the aging field because it's such a new science, right? And we have, like, I feel like a lot of human aging is still a mystery, right? So it's already been valuable for these fields where we have a much deeper knowledge of the mechanisms in the science, like CVD or cancer biology. So we went through the exercise of like, what, it's data sets for two different things, right? Like data sets that, one, can help give us conviction around those mechanisms that work in model organisms, because that'll also translate into humans, right? So sort of translational knowledge. And two, target discovery, novel target discovery in humans, because a lot of, I believe that a lot of the pathways that matter to aging are still undiscovered. And the data sets like the right, genetics and proteomics data sets can help uncover important new targets. And so our approach at BioAge was really to capture molecular changes over the lifetime. Like this is one of the reasons why it's so hard to study human aging in a lab, right? Because it's a 50-year experiment. It's not possible. um, even if you want to do a mouse lifespan experiment, that's the length of a PhD, if you're lucky and it works.
I know, it kills me, the length
Right?
of those experiments, yeah.
Right. And so there were a small number of very forward-looking biobanks that started to collect sample from healthy middle-aged people, close to 50 years ago, and then followed up those individuals throughout the rest of their lives with health records on, how long they lived, the diseases they got as they aged, and also critically, like in our favorite biobank, health span variables. Like every few years, they're looking at walking speed and grip strength and cognitive function over decades. And what we are able to do is go into these samples, these really precious samples that have been conserved for decades, and interrogate them with modern technologies like proteomics. You can look at 7,000 proteins now in a human blood sample, thousands of metabolites, right? And just ask this big sort of data first question: what's changing with aging, of course, but also what predicts the future, right? Like if you look at the molecular pathways in a middle-aged population, what differentiates those individuals who go on to live like 90 plus with really functional muscle from those who don't? And use that as a starting point for all of our science.
Did it shock you that recent studies have shown that the variation in human longevity is basically not genetic, or a very small portion of it is genetic? Was that a surprise? And then, you just mentioned proteomics and metabolomics. I mean, do you have a preference for what data sets you think are going to be the most predictive?
It's not a surprise that common genetic variation doesn't explain much in normal human lifespans, right? You do see that explanatory percentage get higher as you look at more and more extremes. And keep in mind too that if you're going to do a genetics project, then you don't need the trait to be explained by like one or two genes in a very large population. Like a great example is PCSK9, right? Like this is like a blockbuster heart drug, and they found it they found this target by human genetics. There was a family that had rock bottom cholesterol levels, and they had basically PCSK9 knocked out. Like, is that gene that important population wide? No, but is it a wonderful target for the rest of the population? Yes, right. So I'm defending the genetic approach here to discovery. Like I still think that if we look at longevity, exceptional longevity in certain special populations, there will be very interesting targets emerging from that. Even though that's actually not our focus at BioAge, because in order to do that, you need to sequence lots and lots and lots and lots of people, right? So our approach
Yeah.
is a bit different, which is what you touched on, which is looking at these omics that vary over time, that vary over the lifetime, right? Like your genome, whether you're 8 or 80, it's the same mostly. But in contrast, your proteome changes very much as a function of age, and changes in similar ways, right, across populations. So you can actually have a lot of power in the proteome. And so we're really looking at these more sort of common changes that unfold in human populations. And one thing that I especially like about these omics that change through time is that they integrate genomes in the environment, right? Like one pathway we're really excited about at BioAge is apelin signaling. And in our cohorts, middle-aged people who have really active apelin levels, they're living a lot longer, they have functional muscle a lot longer. Now, maybe they were born with genetically high apelin, or maybe they just exercise a lot, or maybe they eat the right foods, right? But whatever it is, right? The molecular intermediary is the apelin, right? And that's the drug target. So I agree that like environment brings a lot to longevity too, and I think that you can see that reflected at the molecular level, and you can like use that to help ID targets as well.
So proteomics, which is really the analysis of all the different proteins in our cells and tissues and the states of those proteins and the level of those proteins, it's very important technology right now. And I think many people are applying this to aging. And then there's metabolomics, which is the small molecules, the metabolites in our tissues and cells and plasma and blood and so on. And you would imagine that the proteomics, because they are the enzymes that do the work in the body, are generating the metabolites. And so I'm wondering if in the end the metabolites will be the thing that we look at, and we don't need to do the proteomics anymore because they predict the metabolites.
Yeah, I think that that's really interesting, right? Like where we are right now is I think we're limited by measurement technologies, right? So if you go into a human blood sample, for example, there's technologies that will let you measure 7,000 proteins, right? But for metabolomics, you can measure and name maybe only around 3,000 metabolites. So in the future, maybe, we'll have like a more, fundamental layer of understanding that we can use to model other ones. But and it's going to be really interesting to see how that evolves. But currently, our favorite modality is probably the proteome. And part of the reason for that is just that we have so much understanding around proteins, right? So if you see a protein signal that's very predictive, then you're not starting in a vacuum of knowledge, right? There's actually a lot of information about this protein. It's probably been mutated in a mouse, and just, it's been described what happens. Maybe even someone tried drugging it for something. And there's this whole like rich literature you can access. I think there are going to be metabolite targets, methylome targets. I think those are harder targets to start with.
Let me take a step back and go back to muscle again. This is obviously one of the major interests with BioAge Labs. And so describe what happens with muscles with age. I just walked up the stairs there and I felt that my legs were a little bit shaky, and I thought, my goodness, what's that all about? And I exercise, but still that was happening.
Yeah, we focus on muscle, and it's one of the universals of aging, right? Like we all lose muscle mass as we age. We all lose muscle function as we age. And this drives a lot of poor outcomes and a lot of suffering. Like it's the number one predictor for frailty and sarcopenia. And when you lose independence, that's when you go into a nursing home. That's when outcomes become very poor. Um, we think it's very important to treat muscle and help extend the health span.
So frailty obviously is a spectrum of different types of aging conditions. Is that something that you can derive patents around and get FDA approval for? Is frailty itself an indication?
That's a great question. I think it should be. I mean, the muscle space is really interesting. I'm not sure if the audience is familiar, but I can give a bit of context there. Like 10, 15 years ago, large pharma did have departments focused on muscle, right? And they were taking things forward like myostatins, like SARMs, selective androgen receptor modulators. And the challenge was they weren't looking even at frailty. They were looking at things like cancer cachexia, which is related, right? Where it's muscle wasting. And the challenge was actually a regulatory challenge. So FDA, acknowledged these were real indications, which they haven't yet for frailty. But the endpoints for these indications to get approval were functional endpoints, which, they kind of have to be for muscle so far. And there were things like the 6-minute walk test. And there's a number of challenges. Like this is such a noisy thing to measure. Like it's a very, center to center variable, administrator to administrator variable, how you feel in one day versus another variable, right? So they're actually very challenging endpoints to hit. And that's one of the important reasons that the field, that muscle departments were shuttered and programs were closed, right? And so I would love frailty to become an indication. And part of that, though, is having like better endpoints, right? Endpoints that are easier to hit in the context of a clinical trial to really see. And we're starting to see a bit of a muscle renaissance just in the last year. One of these old myostatin drugs, the one that Novartis discarded some time ago, got brought into a company called Versanis. This is a great example. And they showed that it could actually help with healthier weight loss in an obesity context. I'm really excited about this because I think that pharma is turning to muscle again. So what's actually really interesting, I'm getting a bit into the weeds on indications now. There's these blockbuster weight loss drugs that didn't used to exist, right? Things that are in the news every week, like Wegovy and Mounjaro. People lose a lot of muscle on those drugs as well. And what companies are starting to realize is that if you pair one of these weight loss agents with a muscle drug, you have the potential for healthier weight loss, but you also have the potential for more weight loss because muscle is metabolically expensive. So it's kind of a, and from a, to return to the earlier point, you know, the regulatory endpoint for these types of drugs is weight loss itself, right? So if you could take your muscle drug, plug it into an obesity drug, and now the bar is a different bar. It's not this very challenging functional endpoint bar. It's a weight loss bar. Suddenly, like there's a lot more development possibility. So there's actually, I would say, a reawakening interest in muscle, which is, I think, good for the field. I thought there's lots of aging mechanisms coming from there. Yeah, it's a bit of a pharma detour, but
No, that's great, actually. I mean, what keeps you awake at night? Is it the day-to-day science, the drug development, or is it this sort of regulatory framework and landscape that you need to navigate?
I mean, it's all plugged together. These are all hard problems, right? Like the science has to work and be meaningful. The drug has to be safe. There has to be a way forward. You know, like no one's going to want to pay for you to develop your drug, to take it into human populations, to show that it works if you can't sell it at the end of the day. So there has to be a very clear understanding of how that's going to work and why there's going to be interest. And that's sort of where we are now, right? Like I would love to see frailty approved as an indication. I think even if we just restrict ourselves to these functional endpoints, if you look at things like wearables, right, like Fitbits or Apple Watches, you might be able to get more robust functional metrics. So I think I think there's a lot of other routes that are, you know, being developed in parallel that might help unlock more possibilities here.
Do you have days where you clearly see you're looking at the data, and of course there's a lot of noise, and aging data has a lot of noise. Is there particular days where you go, there's the signal, there's the target?
I mean, you know what this is like, right? So it's you know, there's sort of the cautious optimism when like one thing works and then like you know, you try it another way or like a different type of experiment altogether and it works again. It's like, oh, that's interesting. And then there's like the rare times when you know, and that's what it looks like when something does work, right? It works over and over again reproducibly with large effect across models, and then you know, then you're set, right? Those are rare. Like those are rare moments, right? A lot of things look promising and you know don't translate across different conditions, or the effect size isn't meaningful enough. But yeah, when you have, I mean, you know, we're still early, right? But there are things that I think are very exciting.
Fantastic. You know, this is I guess this is a question for me. When you mentioned model organisms earlier, and you mentioned mice briefly and things, how much does this factor into your calculations about drug development? I mean, it seems you need to have mouse data to get a patent in the first place. And we know that the mouse models are not optimal by any means in terms of studying functional aging. What's your framework for that, and what's your company's framework for model organisms work?
We do a lot of discovery work in our human data sets, but then yes, you have to, like, even if you believe like you have, you know, Mendelian randomization, like, causal genetic evidence, there's still the separate question of, if I take a sick person and modulate this pathway in the short term, will that suffice to really modify disease, right? So there's still, I think, a really important role for disease models, animal models. The way that we focus on this at BioAge is that we do have an aged colony, and we like to focus on things that naturally go wrong with these animals, you know, with the idea that that's going to be more translatable, things that go wrong in these animals in the same way they go wrong in humans, right? So for example, like old mice on their own will never die of heart disease. Old mice on their own will never develop Alzheimer's. So those, you know, preclinical models we don't really use, right? But old mice like us, they lose muscle mass. They don't run as well in their wheels, right? That's one of the reasons we like muscle aging as well. We believe that we have more access to translational models that are naturally aged, yeah, which we believe a bit more, you know.
You're describing de-risk. Could you just give a very general explanation of what that means?
Yeah, for sure. So in drug development and in biotech, basically everything takes a very long time and every step is more expensive than the prior one. So if your target, you know, that you discovered in mice isn't going to work in humans, it's not going to be efficacious. You want to, like, you know that as soon as you can. You want to know that before spending, you know, $10 million on a phase 2 trial. So if you can de-risk, you know, quote unquote, that sort of means looking at a whole host of things that might make your program, less likely to be true. So there's de-risking from an efficacy perspective, but in de-risking from a safety perspective, right? But just you want to have you want to sort of be able to have as many early looks as you can to sort of be sure that you're on the right path.
So let me just finish by asking you. You mentioned, of course, that the omics, the metabolomics, and the proteomics capture exposure to environment, and this is becoming a big thing, right? That really our entire life experience of the environment, everything we do, everything we eat, the air we're exposed to, the environmental toxins we're exposed to, the exposome is a vast unstudied area, and it's almost completely impossible to think about how to study it. And maybe it doesn't matter, if you develop a drug and it's really, you show it's effective in a certain group of people, and that's fantastic, and that's. But do you think about the need to study diverse groups of people who've been exposed to different risks throughout their life, and even maybe their parents' life, through epigenetic mechanisms?
I guess we're looking for first, those mechanisms that are going to be translated broadly, right? And if you can think of us more as like, in contrast to looking only in inbred black six mice and comparing some young ones to some old ones, even if you were to only go to like one human population, you're still adding vastly more genetic and environmental variability. We have, I guess, right now, sort of a few different ethnicities covered, but it's more like 10,000 people, not millions of people, right? So there probably is a lot more to discover as you add on more diversity. Especially for particular paradigms, right? Like there are some people who are incredibly stress resistant. There are some people who, you know, eat a terribly unhealthy diet, but live really long and healthy, right? So I think there's a lot of different types of discovery paradigms that could be fruitful. Yeah.
I presume you're very conscious of gender differences as well in drug development, and what we can do there to improve that situation.
That's been a big one in aging biology, right? It's frustrating. It'd be nice if things were to worked really well in both, but but so far there have been quite pronounced differences, yeah, in genders. Yeah, that's an interesting one.
Well, I've got another final question. Where do you see BioAge in 10 years' time, and where do you see the aging field in 10 years' time?
Yeah, that is a very hard question, but but I would like us to have like an approved drug, ideally a couple, right? Like I would like us to be testing other mechanisms and the field as a whole, you know, I think we're in an interesting spot right now, as we were discussing earlier. There's sort of more activity in terms of translation than there's really ever been before. It's again still early days, right? Like you can draw a map of the clinical pipeline for aging, and if you look at things that are near IND or in human testing, there's maybe 20 or so things. That's a big number. Until you look at like the pipeline of a Novartis or a Lilly, where they have like 120 different things in the clinic, right? And that's just one company. So there's still a lot of room to grow, and I think there's a lot of very exciting mechanisms that are in or near human testing. So I think the field is going to be changing rapidly, like expanding rapidly.
Is that a problem of academia not coming up with new interventions? Is that a problem of not enough aging companies yet?
All of the above. And really like funding for ideas too coming out of aging biology, right? Like it's still a new field to biotech. It's still a new field for pharma. Once we start to see some of the first successes in terms of molecules that make it over the line, I think we're going to see a lot more spending in this area, right? Which hopefully, like I believe there's a lot more mechanisms to discover, but I also believe that like there's are a lot of things that are ready to be translated that haven't been yet, right?
In thinking about the future, what if we get outside of ourselves, outside of the aging bubble? What do you expect the layperson, or anyone who's experiencing aging and seeing horrific things happen within their families with Alzheimer's and cancer and so on? What would you hope that they start to see or hear about this field?
Well, in terms of what they see, like right now, as you grow older and you think about what medicines are really available for preventing, disease instead of treating disease. And there's not many, right? Like I would put statins in that category. Most people after a certain age start taking those. But I think that that's really the ultimate potential of these aging medicines, right? Like I would like to see that set of options widen, where they actually have that potential to delay disease. And at the same time, more awareness right now, right? Like we discussed muscle aging, and that's in a way like that's horrible. But I want to keep my parents out of a nursing home. Like that's a scary outcome too. That's a terrible place to be. And just recognizing that there are ways, there is science that can help, and
Yeah.
there should be more attention focused on this, so that I think that society should sort of shift focus that way. I think now when people are past a certain age, you go to the doctor, right? They're like, oh, sorry, you're old, right? And I think we can do better. Yeah.
Kristen Fortney, it's been an absolute delight. Thank you so much. You're a real pioneer in this field in taking us towards translation. And you're also a very generous communicator of what you do and what your company does. So thank you for taking this time with us today.
Thanks so much, Gordon. Really enjoyed it.
Thank you for listening. Please subscribe, share, and give us a 5-star review on Apple, Spotify, or wherever you get your podcasts. We're Not Getting Any Younger Yet is produced by Vital Mind Media. The Buck Institute's very own Robin Snyder is the executive producer. Wellington Bowler is right next to me here, directing the recordings. Stella B is behind the scenes, ready to debrief when we wrap. And the esteemed Sharif Izzat weaves the show together for you. If you're listening to the podcast, you'll know that there has never been a more exciting time in the research on aging. Discoveries in the lab are moving to the clinic to help us all live better, longer. The Buck Institute depends on the support of people like you to carry on our breakthrough research. Please visit us at buckinstitute.org to learn more and to donate.